the necessary partnerships. If the topmost rung on one strand was made of guanine, then you would know that the topmost rung on the matching strand must be cytosine. Work your way down the ladder through all the nucleotide pairings, and eventually you would have the code for a new molecule. That is just what happens in nature, except that nature does it really quickly, in only a matter of seconds, which is quite a feat. Most of the time, our DNA replicates with dutiful accuracy, but just occasionally, about one time in a million, a letter gets into the wrong place. This is known as a single nucleotide polymorphism, or SNP, familiarly known to biochemists as a SNP. Generally, these SNPs are buried in stretches of non-coding DNA and have no detectable consequence for the body, but occasionally they make a difference. They might leave you predisposed to some disease, but equally they might confer some slight advantage. More protective pigmentation, for instance, or increased production of red blood cells for someone living at altitude. Over time, these slight modifications accumulate both in individuals and in populations, contributing to the distinctiveness of both. The balance between accuracy and errors in replication is a fine one. Too many errors and the organism can't function, but too few and it sacrifices adaptability. A similar balance must exist between stability and innovation in an organism. An increase in red blood cells can help a person or group living at high elevations to move and breathe more easily, because more red cells can carry more oxygen. But additional red cells also thicken the blood. Add too many, and it's like pumping oil, in the words of Temple University anthropologist Charles Weitz. That's hard on the heart. Thus, those designed to live at high altitude get increased breathing efficiency, but pay for it with higher risk hearts. By such means does Darwinian natural selection look after us. It also helps to explain why we are all so similar. Evolution simply won't let you become too different, not without becoming a new species anyway. The 0.1% difference between your genes and mine is accounted for by our SNPs. Now, if you compared your DNA with a third person's, there would also be 99.9% correspondence, but the SNPs would, for the most part, be in different places. Add more people to the comparison, and you will get yet more SNPs in yet more places. For every one of your 3.2 billion bases, somewhere on the planet there will be a person or group of persons with different coding in that position. So not only is it wrong to refer to the human genome, but in a sense we don't even have a human genome. We have 6 billion of them. We are all 99.9% the same, but equally, in the words of the biochemist David Cox, you could say all human beings share nothing, and that would be correct too. But we have still to explain why so little of that DNA has any discernible purpose. It starts to get a little unnerving, but it does really seem that the purpose of life is to perpetuate DNA. The 97% of our DNA commonly called junk is largely made up of clumps of letters that, in Matt Ridley's words, exist for the pure and simple reason that they are good at getting themselves duplicated. Most of your DNA, in other words, is devoted not to you, but to itself. You are a machine for the benefit of it, not it for you. Life, you will recall, just wants to be, and DNA is what makes it so. Even when DNA includes instructions for making genes, when it codes for them, as scientists put it, it is not necessarily with the smooth functioning of the organism in mind. One of the commonest genes we have is for a protein called reverse transcriptase, which has no known beneficial function in human beings at all. The one thing it does do is make it possible for retroviruses such as HIV to slip unnoticed into the human system. In other words, our bodies devote considerable energies to producing a protein that does nothing that is beneficial and sometimes clobbers us. Our bodies have no choice but to make it because the genes order it. We are vessels for their whims. Altogether, almost half of human genes, the largest proportion known in any organism, don't do anything at all, as far as we can tell, except reproduce themselves. All organisms are in some sense slaves to their genes. That's why salmon and spiders and other types of creature, more or less beyond counting, are prepared to die in the process of mating. The desire to breed, to disperse one's genes, is the most powerful impulse in nature. As Sherwin B. Newland has put it, empires fall, ids explode, great symphonies are written, and behind all of it is a single instinct that demands satisfaction. From an evolutionary point of view, sex is really just a reward mechanism to encourage us to pass on our genetic material. Scientists had only barely absorbed the surprising news that most of our DNA doesn't do anything, when even more unexpected findings began to turn up. First in Germany and then in Switzerland, researchers performed some rather bizarre experiments that produced curiously unbizarre outcomes. In one, they took the gene that controlled the development of a mouse's eye and inserted it into the larva of a fruit fly. The thought was that it might produce something interestingly grotesque. In fact, the mouse eye gene not only made a viable eye in the fruit fly, it made a fly's eye. Here were two creatures that hadn't shared a common ancestor for 500 million years, yet could swap genetic material as if they were sisters. The story was the same wherever researchers looked. They found that they could insert human DNA into certain cells of flies, and the flies would accept it as if it were their own. Over 60% of human genes, it turns out, are fundamentally the same as those found in fruit flies. At least 90% correlate at some level with those found in mice. We even have the same genes for making a tail, if only they would switch on. In field after field, researchers found that whatever organism they were working on, whether nematode worms or human beings, they were often studying essentially the same genes. Life, it appeared, was drawn up from a single set of blueprints. Further probings revealed the existence of a clutch of master control genes, each directing the development of a section of body, which were dubbed homeotic, from a Greek word meaning similar, or Hox genes. Hox genes answered the long, bewildering question of how billions of embryonic cells, all arising from a single fertilized egg and carrying identical DNA, know where to go and what to do. That this one should become a liver cell, this one a stretchy neuron, this one a bubble of blood, this one part of the shimmer on a beating wing. It is the Hox genes that instruct them, and they do it for all organisms in much the same way. Interestingly, the amount of genetic material and how it is organized doesn't necessarily or even generally reflect the level of sophistication of the creature that contains it. We have 46 chromosomes, but some ferns have more than 600. The lungfish, one of the least evolved of all complex animals, has 40 times as much DNA as we have. Even the common newt is more genetically splendorous than we are, by a factor of 5. Clearly, it is not the number of genes you have that matters so much as what you do with them. This is a very good thing, because the number of genes in humans has taken a big hit lately. Until recently, it was thought that humans had at least 100,000 genes, possibly a good many more, but that number was drastically reduced by the first results of the Human Genome Project, which suggested a figure more like 35,000 or 40,000 genes, about the same number as are found in grass. That came as both a surprise and a disappointment. It won't have escaped your attention that genes have been commonly implicated in any number of human frailties. Exultant scientists have at various times declared themselves to have found the genes responsible for obesity, schizophrenia, homosexuality, criminality, violence, alcoholism, even shoplifting and homelessness. Perhaps the apogee or nadir of this faith in biodeterminism was a study published in the journal Science in 1980, contending that women are genetically inferior at mathematics. In fact, we now know, almost nothing about you is so accommodatingly simple. This is clearly a pity in one important sense, for if you had individual genes that determined height or propensity to diabetes or to baldness or any other distinguishing trait, then it would be easy, comparatively easy anyway, to isolate and tinker with them. Unfortunately, 35,000 genes functioning independently is not nearly enough to produce the kind of physical complexity that makes a satisfactory human being. Genes, clearly, therefore, must cooperate. A few disorders, hemophilia, Parkinson's disease, Huntington's disease, and cystic fibrosis, for example, are caused by lone dysfunctional genes. But as a rule, disruptive genes are weeded out by natural selection long before they can become permanently troublesome to a species or population. For the most part, our fate and comfort, and even our eye color, are determined not by individual genes, but by complexes of genes working in alliance. That's why it is so hard to work out how it all fits together, and why we won't be producing designer babies anytime soon. In fact, the more we have learned in recent years, the more complicated matters have tended to become. Even thinking, it turns out, affects the way genes work. How fast a man's beard grows, for instance, is partly a function of how much he thinks about sex, because thinking about sex produces a testosterone surge. In the early 1990s, scientists made an even more profound discovery when they found they could knock out supposedly vital genes from embryonic mice and still see the mice often not only born healthy, but sometimes actually fitter than their brothers and sisters who had not been tampered with. When certain important genes were destroyed, it turned out, others were stepping in to fill the breach. This was excellent news for us as organisms, but not so good for our understanding of how cells work, since it introduced an extra layer of complexity to something that we had barely begun to understand anyway. It is largely because of these complicating factors that cracking the human genome came to be seen almost at once as only a beginning. The genome, as Eric Lander of MIT has put it, is like a parts list for the human body. It tells us what we are made of, but says nothing about how we work. What's needed now is the operating manual, instructions for how to make it go. We are not close to that point yet. So now the quest is to crack the human proteome, a concept so novel that the term proteome didn't even exist a decade ago. The proteome is the library of information that creates proteins. Unfortunately, observed Scientific American in the spring of 2002, the proteome is much more complicated than the genome. That French geneticist Jacques Monod put it, only half in jest, anything that is true of E. coli must be true of elephants, except more so. Every living thing is an elaboration on a single original plan. As humans, we are mere increments, each of us a musty archive of adjustments, adaptations, modifications, and providential tinkerings stretching back 3.8 billion years. Remarkably, we are even quite closely related to fruit and veg as the chemical functions that take place in you. It cannot be said too often, all life is one. That is, and I suspect will forever prove to be, the most profound true statement there is. And 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 true statement. Part 6. The Road to Us. Descended from the apes. My dear, let us hope that that is not true. But if it is, let us pray that it will not become generally known. Remark attributed to the wife of the Bishop of Worcester, after Darwin's theory of evolution was explained to her. Chapter 27. Ice Time. I had a dream which was not all a dream. The bright sun was extinguished, and the stars did wander. Byron, Darkness. In 1815, on the island of Sumbawa in Indonesia, a handsome and long quiescent mountain named Tambora exploded spectacularly, killing 100,000 people with its blast and associated tsunamis. No one living now has ever seen such fury. Tambora was far bigger than anything any living human has experienced. It was the biggest volcanic explosion in 10,000 years, 150 times the size of Mount St. Helens, equivalent to 60,000 Hiroshima-sized atom bombs. News didn't travel terribly fast in those days. In London, the Times ran a small story, actually a letter from a merchant, 7 months after the event. But by this time, Tambora's effects were already being felt. 240 cubic kilometers of smoky ash, dust, and grit had diffused through the atmosphere, obscuring the sun's rays and causing the earth to cool. Sunsets were unusually but blearily colorful, an effect memorably captured by the artist J.M.W. Turner, who could not have been happier. But mostly the world existed under an oppressive, dusky pall. It was this deathly dimness that inspired Byron to write the lines just quoted. It was a dreadful year, almost certainly the worst for farmers in modern times. Yet globally, the